In this video, we're going to talk about cell theory, and more specifically, what the cell theory actually is, the evidence for the cell theory, and the technology needed to be able to collect that evidence. So the cell theory is three main points. All living things have cells, so that's important. They have to actually have cells, but also more importantly is that the cell for all living things is the structural and functional unit of living things. So if you have no cells, that means living things can't operate. They help them make energy, they help them be able to digest food. All functions in living things are performed by cells. So without cells, there are no living things. That's the second. So the first and second are kind of connected. And the third, more or less law of the cell theory, or the statement of the cell theory, is that cells come from pre-existing cells. Right? So what that means is that if we have one cell that can double and make two cells, and a cell can't just come out of nowhere, it can't do spontaneous generation, it'll always come from a pre-existing cell. So these three are the statements of the cell theory. And what you need to know is what evidence we have for that, so the evidence for the actual cell theory. And for that first statement, so the first statement was that all living things have cells. For that, we have evidence because cells, and so red and red go together, cells can be seen with the microscope. And we've checked more or less all living things, and everything that's living, we can find those cells within them. So that's our evidence for that all living things have cells. We use a microscope, and we can actually look at the cells within that living thing. Now, for the second point, that the cell is the structural and functional unit of living things, the evidence we have, which is down here, is that if a cell or too many cells die, then so does the living thing. So what that means is that cells provide that structure and function, and the cells go, we go as well. So that's evidence that cells basically provide us with the function we need to survive. So that's the evidence. We know that if cells die, we die, and that helps us support that cells are a structural and functional unit of living things. So for the last one, cells come from pre-existing cells. There's two parts. First of all, we definitely have done the Lewis Pasteur experiment. Now, Lewis Pasteur experiment helped disprove spontaneous generation because it could show that things just don't come from nothing, so they have to come from pre-existing cells. And also we've been able to observe mitosis and meiosis. That's cell division. So we've actually been able to see it with a microscope. We're able to observe those two processes. So these are the evidences. So these three here are the evidences for the three statements of cell theory. And what technology have we needed to be able to actually find those evidences? That's what we're going to talk about next. So for example, if we want to be able to see living things on a microscope, obviously we need to have a microscope. So the light microscope was one big invention that helped us be able to actually identify the importance of cells. And there was a light microscope and electron microscope. We'll talk about those more in a second. Both of them were important to be able to actually see cells because they're so small, they're so microscopic. Better staining techniques. We said that was something that you get that image that has the different shades. We'll look at one of those images soon, and those different shades are done with staining techniques. And also better lenses. So not just better microscopes or new microscopes, but the lenses, if they're more powerful, we have more magnification, which means we can see smaller things. So these were some technological advances we needed to be able to actually give evidence for the cell theory. Now, the last one here is just some history. Remember, you need to know these names. So Robert Hooke and Robert Brown. And this scale starts at 1600, ends at about 2000, roughly nowadays. First thing you should know is just before 1600, so about 1590, that's when the first light microscope was invented. That's important to be able to make all those discoveries we've been able to make afterwards, because without the microscope we can't see cells. After the next important person was Robert Hooke. And what Robert Hooke did, he looked at a cork cell, or a cork, not just a cork cell, but a cork, and he saw all these little mini cells, and he coined the term cell, and that was in 1653. Afterwards was 1673. Remember, that was Leeuwenhoek, and Leeuwenhoek, he was the person who actually saw the first very small microscopic thing. So Robert Hooke just looked at a dead animal, a plant cell, but Leeuwenhoek actually saw living microscopic animals in water. So he was the first person to see something that's microscopic and that's living in water. And then in 1833, another person called Robert Brown came. Robert Brown was the person who identified the nucleus in plants. And the invention of the electron microscope was in 1931, and as we'll talk about in a second, that helped us to basically be able to see many more organelles within the cell, which helped us improve our understanding of the cell. Now we're going to look at an actual cell, and we're going to talk about all different types of organelles the cell actually has. We're going to talk about the function of those organelles, the name of the organelles, and also how the structure and function are connected. So we'll start with just basically mentioning that this is a plant cell, and by the end I'll make sure we go through what the difference between plant and animal cells are. This here is meant to be a plant cell. Alright, so we have a couple of organelles that are only present in plants, and a couple of them which are present in both animals and plants. But I'll go for all of them first. The one that's in the middle here is the nucleus. The nucleus contains DNA, and that DNA is important for something called protein synthesis, which basically makes protein. So that's what they're for. So DNA will help you indirectly make protein, and these proteins are very important. Skin is made of proteins, enzymes are proteins, a lot of things in the cell are proteins. So the DNA is ultimately the instruction manual for life, because it allows us to make these proteins, which we need to be able to survive. That's DNA, and that's found in the nucleus. So the nucleus contains DNA, and that DNA will help make proteins, and the nucleus is often called the control center because it will regulate what is being made in the cell. Next to it we've got the chloroplast, which is right here. The chloroplast is only found in plant cells, and it does photosynthesis, which means it allows it to be able to make glucose, which is a sugar, and that glucose will then be able to be made into energy. We'll talk about that in a second. Right, so chloroplasts make glucose, only found in plant cells, and do so through a chemical reaction or a chemical chain of reactions called photosynthesis. We have also the large vacuole. The large vacuole can be found in plant cells. The animal cells have small vacuoles. These vacuoles, they're more or less storage site for lipids. Lipids are fats, so we can store your fats in those large vacuoles. Your cell walls, again, this is something that only plant cells have, bacteria cells too, but not animal cells. Cell wall gives shape, protection, and structure to a cell, and a animal cell can't have it because it has to be flexible, it has to be able to move around. A cell wall is very rigid, so it would be too rigid for an actual animal cell. We also have the Golgi Body. We'll talk about the Golgi Body in a sec, but actually before we do go there, I want to talk about the ribosomes first. Ribosomes are right here, so these little dots in the cytoplasm, and also these little dots here on the roughy-R. Ribosomes produce proteins. We talked about the nucleus. It has the instruction manual for proteins, and that instruction manual will then eventually end up at the ribosomes, and at the ribosomes, that's where proteins will actually be made. Right, so ribosomes and your nucleus will work together to help you make proteins, and those proteins were very important. Now, roughy-R, that's going to be joined to your actual nucleus right here. Roughy-R is basically just a place where that RNA, which will be made in the nucleus but then delivered out to the ribosomes, can travel in. So roughy-R is a more or less a highway system that connects the nucleus and can make sure that a lot of proteins are made. Right? Roughy-R is full of ribosomes, and those ribosomes will make those proteins. So remember, roughy-R, rough because those ribosomes are spotted all over the actual endoplasmic reticulum. That's what ER stands for. And roughy-R is responsible for making proteins because it has lots of ribosomes attached. Now we also have smooth-ER, which is joined to the roughy-R, but it doesn't have ribosomes. Hence it's called smooth because it doesn't have those dots. And it is responsible for drug detoxification. So for example, alcohol gets detoxified there. Mitochondria help produce ATP. So we said that your photosynthesis makes glucose, so chloroplasts make glucose, and mitochondria will break those glucose down to make ATP. We also have your cell membrane. The cell membrane surrounds every cell, and what that does, it gives a cell shape. It also is semi-selective. It does semi-selective transport, which means it can decide what goes in and what goes out. It can control that, which is good. And it's obviously very important. All cells have cell membranes. Lysosomes are there for often recycling. Old organelles, when they die down, they have to be deassembled and made into the new ones, and lysosomes help you digest and recycle those old organelles. And then we have the Golgi body, which I said we'll talk about now. So the Golgi body is this kind of structure here. We'll talk about the actual how it looks on a microscope in a minute. But the Golgi body is there to package and modify proteins. So proteins get made at the rough ER, and then they're actually sent from the rough ER. They are sent to the Golgi body. From there, they're modified, changed, and then they're sent on out of the cell if they are made at the rough ER. So proteins can be made in the cytoplasm and the liquid inside, or on the rough ER, and then they're wanted for transport outside of the cell. So the Golgi body is a packaging center, and it modifies proteins as well. Alright, so that's basically your main structures. The only one I didn't talk about yet is cytoplasm. Cytoplasm is that liquidy component inside of a cell. It's basically anything that's inside of your cell membrane is part of the cytoplasm, and that has mostly made up of water, dissolved different nutrients and chemicals. That's where all the chemical reactions take place. So that's what cytoplasm is. Now, you need to know which parts are part of a plant cell and which are part of the animal cell. I'm going to basically underline, so I'm going to use this color right here to show which of these organelles are only found in plant cells. The other ones are also found in animal cells. So chloroplast is plant only. So is the cell wall, and so is the large vacuole. Animals do have vacuoles sometimes, but they're small vacuoles. And all of the rest of the organelles, both plant and animal cells have. But those three I mentioned there are found only in plant cells. The electron, so the next, what I'm going to talk about next is which ones of those you can see on the light microscope, the less powerful one, and for which ones you need the electron microscope, the more powerful one. So the first couple I mentioned are all that you need the electron microscope, light microscope for. So you can see the cell wall with the light, so I'll say L for light. You can see also the large vacuole with the help of your light microscope. You don't need to have electron microscope. You can see the nucleus with your light microscope. And you can see the chloroplast with your light microscope. All the other ones, the ones I didn't mention, they all have to have electron microscope to be able to actually identify and see them. So large vacuole, cell wall, nucleus, and chloroplast are the ones we see with the light microscope. For all the other organelles, the ones I didn't mention just now, we need the electron microscope to be able to see them. And last thing, we have these four images. These are images of electron microscope diagrams of different organelles. The first one here is your mitochondria. So let's write M for mitochondria. This shape is always your mitochondria. So you have that beanish shape, and you have some folding happening. That's to increase surface area for chemical reactions. When it comes to your chloroplast, this is the chloroplast. It will always look like that, and it has stacks. Those stacks are going to be filled with chlorophyll, and light will hit those stacks to do photosynthesis. Right, so that's always what chloroplast will look like. This here is actually your Golgi body. It has that unique shape also to increase surface area. And the big thing in the middle of an actual cell is going to be your nucleus. And those are 4 examples of organelles of how they actually look like under the microscope, but you need to make sure you know more than that.